The team of scientists from the Department of Biological Sciences have made a significant discovery that will have a huge impact on research into eye development and the possibility of actually creating an eye in the laboratory. I've been along to find out more about this groundbreaking research from the team leaders, Professor Liz Jones, but first we hear from Professor Nick Dale.
We are going to know enough about the development of tissues, the key signalling processes involved, the genes involved. Our work gets us a little bit along the way. We've discovered a very important and unpredictable part of that jigsaw, but there are other key parts that need to be filled in before we can do that.
We can see that the eyes that form often have extensions to the midline of the animal, which is where the nervous system forms. So that would suggest that they could be connected electrically to the brain. But we've not done experiments that would look at that, and in fact the animals that we use in experimentation don't live as long as those experiments could be performed. People thought that eyes could only develop in the head, and what we can do is we can target these molecules to different parts of the embryo and ask whether they can make eyes in other parts of the embryo apart from the head. And we could show that in small numbers of cases that we could see eyes with lenses actually in the tails of the animals, not in the head at all. So that dispelled the view that eyes can only be formed in the head.
So once you'd had this eureka moment, what happened then?
Well, we then spent an awful long time firming it up. So we could see that there were extra eyes, and we could see that by the fact that you could see pigmentation, which was indicative of eye structure. So we looked at the eyes to see whether they had layers associated with eyes, as in neuronal layers, which they did have.
There was the excitement of the initial observation, but then of course we weren't sure whether this was really important or not, so we had to make further experiments to follow up. So a very important experiment was not just to overexpress our protein and gene of interest and make more eyes, but to ask an even more harder question: what would happen if we knock down the expression of that protein in the real animal? Would we prevent the eye from occurring? And we managed to do that, and I think that was the most exciting.
It's going to take a lot of time because that was just, the first steps. We just discovered these mechanisms, but we don't really know exactly how it does work. We have to identify which cell do release the ATP, and I know it goes from these cells to switch on the genes which are involved in the eye development. So it's going to take, I think, a few more years and work. The first time when Liz injected the gene of interest, and we've seen these first ectopic eyes, where at this point we didn't know it was an ectopic eye, but it was an ectopic structure. That was kind of surprised us, and we just all looked down under the microscope and we were just there, wow, that is going to be something. But that was the real first time. I remember that day very well.
This type of research takes a long time. It takes quite a lot of money. It takes a long time to do. The experiments are very painstaking. They involve molecular biology, functional analysis, anatomical analysis, and so it would take years to be able to understand all the events that take place in eye development. If one understands what the process is that triggers a whole network of molecules which are involved in eye development, then long term there is the potential to switch stem cells, which are these undifferentiated cells. And that might mean ultimately that one can make the eye in the dish by converting cells which are not normally eye cells into eye cells. But that's a long way down the track.
This was a very serendipitous accident, but I think this sort of thing can happen where you bring different approaches together, so that you sort of step outside your immediate scientific furrow, try to borrow things from outside, and sometimes good things happen and unexpected results occur which are very important.
We were not expecting anything to do with eye phenotypes, and it was a real surprise, which is one of the delights of science, if you like, in that you get the unexpected and then you follow that line of experimentation.